Good day, I'm Dr. Raghav Sundar. I'm a consultant medical oncologist at the National University Cancer Institute of Singapore. I'm also a clinician scientist, part of the Singapore Gastric Cancer Consortium. Together with Professor Patrick Tan, Professor Akira Tsuburaya, and Professor Takashi Oshima, along with the investigators from the SAMIT trial, I'm pleased to present to you data that has recently been published in the journal Gut. Paclitaxel is a chemotherapeutic agent that has activity in gastric cancer. However, the activity is not universal, with several tumors demonstrating resistance. Therefore, there is a need to identify biomarkers that predict for paclitaxel benefit. Studies like the TCGA use unsupervised clustering techniques. These methods analyze and cluster unlabeled datasets to uncover relationships or patterns within the data without the need for human supervision. Unsupervised learning is not used to predict outcomes. In our study, our goals was to specifically identify predictive biomarkers of resistance to chemotherapy. Therefore, we used a supervised machine learning algorithm. Supervised learning uses labeled datasets that are used to train algorithms. In our study, we used the random forest machine learning algorithm. Random forest is based on the ensemble learning concept in machine learning. In ensemble learning, several models are implemented to accomplish a single task. Each tree is grown by using a bootstrap sample of the dataset, and only a random subset of the original variables is examined at each node. Random forests return a prediction from a collection of classification trees. A voting system is utilized to adjudicate the final class label. Multiple iterations of the random forest algorithm are run to generate a variable importance analysis. SAMIT was a randomized phase 3 trial conducted in Japan. The study tested two separate hypotheses. First, does the addition of sequential paclitaxel to oral 5-FU, such as S1 or UFT, improve survival over S1 or UFT alone? The second hypothesis was whether UFT was non-inferior to S1. In our study, the goal was to specifically identify predictive biomarkers of resistance to paclitaxel. From a total of nearly 1,500 patients that were involved in the clinical trial, nearly 500 samples were profiled. We took advantage of the unique 2x2 factorial design of the SAMIT trial. Essentially, each arm of the study could function as an individual cohort of patients for training and validation. As SAMIT was a randomized phase 3 trial, two paclitaxel treated cohorts, Pac-S1 and Pac-UFT, and two paclitaxel untreated cohorts, S1 and UFT alone, were formed. We designed a customized nanostring panel of approximately 500 probes to study various features of the tumor which we believed would contribute to paclitaxel sensitivity. The panel consisted of genes involved in spindle assembly checkpoint, chromosomal stability, mitosis, DNA damage repair, and immunogenic cell death. We found 19 genes contributed maximally to the survival difference in the training cohort. These 19 genes form the random forest gene signature. Next, we applied this model to the other arms of the study. We also applied the signature on a separate, independent dataset of patients with metastatic gastric cancer treated with paclitaxel and ramucirumab in a phase 2 trial from South Korea. We were able to demonstrate that the classifier correctly identified better survivors in the paclitaxel containing cohorts, Pac-UFT and Pac-RAM, but there was no difference in survival between the two groups in the non-paclitaxel containing arms. Test of treatment interaction with the classifier was statistically significant. Hence, we were able to demonstrate that this signature functioned as a robust predictive biomarker for paclitaxel sensitivity. The strength of this biomarker is the ability to validate robustly in internal and external test cohorts. In summary, we use machine learning techniques on one of the largest gastric cancer trials to date to identify a gene signature representing the first predictive biomarker for paclitaxel benefit. Given the rising utilization of taxanes in gastric cancer, particularly in the adjuvant and perioperative setting, this biomarker may be a guide in identifying patients who benefit from taxane-based therapy following further validation in prospective trials. We would like to thank the patients who have magnanimously contributed their tissue samples and data to further the science.